In 2010, German geneticists sequenced DNA from a bone fragment found in a Siberian cave, a finger bone, possibly belonging to a child. Name me any other biological species discovered from a finger bone and 3 teeth. There isn't one, only this one. The scientists expected to find a Neanderthal or an early Homo sapiens. Instead, they found something third, a creature close enough to us to interbreed and produce offspring, and distant enough to be a separate species entirely, unknown to science. The Denisovans became the only species in the history of paleoanthropology identified exclusively through molecules. We knew their genome before we knew their face. We knew they lived across Asia, Siberia, and possibly the tropics before we found a single one of their skulls. And in the 15 years since that discovery, every new year has rewritten what we thought we knew about them. Last year, we finally found their face. This year, their traces in the genome of creatures who lived 400,000 years ago. The Denisovans are gone, but they never left. Their DNA is working inside living people right now, protecting, adapting, surviving. This is their story, and in part, it is yours. Before we go any further, please subscribe to this channel. Your support really helps us to continue this work. Thank you. There is a concept in genetics called a ghost lineage. It refers to a branch of the evolutionary tree that you know must have existed because its effects are visible in the organisms that descended from it, but for which you have no direct fossil evidence. A ghost lineage is an inference, a shadow on the wall of the cave cast by a fire you cannot see. For most of the 20th century, the story of human evolution was told in terms of creatures we had bones for: Australopithecus, Homo erectus, Homo heidelbergensis, the Neanderthals. Each of these species was known from physical remains: skulls, teeth, limb bones, the durable architecture of bodies that had once moved through a world we were trying to reconstruct from their remnants. The story was incomplete, as such stories always are, but it had a physical foundation. You could put your hands on the evidence. The discovery of the Denisovans changed the epistemology of human evolution. For the first time, a member of the human family was identified not from its bones, but from its biochemistry, from the sequence of nucleotides in its DNA, extracted from a fragment of bone so small that it could sit comfortably in the palm of your hand. The Denisovans were not deduced from a skull. They were read from a molecule. And that fact, that we knew their genetic identity before we had any idea what they looked like, has given the entire field of paleoanthropology a quality that it did not previously possess. It has made human evolution feel, in the most literal sense, like a mystery story. One in which the key witness left a written confession but no photograph. The story begins, as so many extraordinary scientific discoveries do, with something that looked completely routine. In 2008, Russian archaeologists excavating Denisova Cave in the Altai Mountains of southern Siberia, a region that had already yielded Neanderthal material and was well known in the paleontological literature, recovered a small fragment of a finger bone from sediment layers in the cave's east gallery. The fragment was catalogued as Denisova 3. It showed no obvious features that would distinguish it from other hominin bone fragments of the Pleistocene, and it attracted no particular attention at the time of its excavation. It was the kind of find that goes into a storage box and waits. 2 years later, it was sent to Svante Pääbo's laboratory at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, for DNA analysis. Pääbo's group had spent the preceding decade developing and refining the techniques of ancient DNA extraction and sequencing. Techniques that had made possible the first draft sequence of the Neanderthal genome, published earlier in 2010. The expectation was that Denisova 3 would yield Neanderthal DNA. The Altai region was known Neanderthal territory. The cave had already produced Neanderthal material. The simplest hypothesis was that this was one more Neanderthal. The DNA said otherwise. The mitochondrial genome, the circular DNA found in the cellular organelles called mitochondria, which is inherited exclusively through the maternal line and serves as a particularly useful marker for lineage identification, was unlike anything in the existing database. It was not modern human. It was not Neanderthal. It was something else, and it was something else in a specific and quantifiable way. The mitochondrial DNA of Denisova 3 diverged from that of modern humans approximately 1 million years ago, roughly twice as long ago as the divergence between modern humans and Neanderthals. This was not a variant of a known lineage. This was a previously unknown branch of the human family tree, one that had been living and presumably flourishing in Asia while our ancestors were evolving in Africa and Neanderthals were spreading across Europe. The paper announcing this discovery was published in Nature in March of 2010. It caused a sensation in the paleoanthropology community, which is not a community given to sensationalism, for one very simple reason. This was not how species were discovered. Species were discovered from bones. You found the skeleton, or enough of it to characterize the organism's anatomy. You compared it to what was known, and if it was sufficiently different, you named it and placed it in the tree. The Denisovans broke this model completely. They were discovered from a molecule. They were named, informally, after the cave before anyone knew what they looked like. They entered science as a genetic signature before they entered it as a physical form. And the genetic signature, as it turned out, was the most interesting part. Because alongside the mitochondrial genome, Pääbo's group extracted and sequenced the nuclear genome of Denisova 3. The DNA in the cell nucleus that is inherited from both parents and carries the vast majority of the genetic information that makes an organism what it is. And in that nuclear genome, they found something that no one expected. They found evidence of interbreeding, not between Denisovans and modern humans in Siberia, where the cave was located. The signal was elsewhere entirely. It was in the genomes of living people in Melanesia and Australia. Think about the geography for a moment. The cave where the Denisovan finger bone was found is in the Altai Mountains, in what is now southern Russia, more than 4,000 miles from the nearest Melanesian island. And yet the strongest genetic signal of Denisovan ancestry in the modern world is not in Siberia. It is in populations on the other side of Asia, in Papua New Guinea, in the islands of Wallacea, in the Aboriginal populations of Australia. Some Melanesian populations carry up to 6% Denisovan DNA in their nuclear genomes. This is not a trivial amount. It is comparable to, and in some populations greater than, the Neanderthal DNA present in non-African modern humans. Someone, somewhere, at some time between 60 and 80,000 years ago, when the ancestors of modern Melanesians and Australians were making their way through Southeast Asia on their journey south, encountered Denisovans. And they did not simply encounter them, they had children with them. This is the image that should sit in your mind as you follow this story. Not the sterile laboratory in Leipzig where the DNA was sequenced. Not the cave in Siberia where the bone was found. The image that matters is something else entirely. Two groups of people, different enough to be separate species, similar enough to recognize each other as something like kin, meeting somewhere in the forests or along the coasts of what is now Southeast Asia, or the islands between Asia and Australia, tens of thousands of years ago. The specific circumstances of that meeting are lost, whether it was a single event or many, whether it was peaceful, we cannot know. What we know is that it produced offspring who survived to adulthood, who had children of their own, and whose descendants eventually became the ancestors of millions of people alive today. That meeting is in your DNA. If you have Melanesian, Australian, Aboriginal, or certain Southeast Asian ancestry. It happened. It mattered. And we have only known about it for 15 years. Now here is where the story takes its first unexpected turn. Because the genetics of Denisovan ancestry, as researchers have studied it more carefully, does not tell a simple story of one ancestral population of Denisovans who encountered modern humans once in Southeast Asia. The genetics tells a story of multiple populations, of different groups of Denisovans, genetically distinct from each other, who interbred with modern humans at different times and in different places. This is not a single chapter. It is a book. The evidence for this comes from comparing the pattern of Denisovan DNA in different modern human populations. If there had been only one interbreeding event or one source population of Denisovans, the Denisovan DNA in different modern human groups should look broadly similar. It does not. The Denisovan ancestry in Papuan and Australian populations is genetically different in measurable ways from the Denisovan ancestry in East Asian populations. And the Denisovan ancestry in the Tibetan plateau population is different again. This pattern of differentiation implies that modern humans encountered not one Denisovan population, but several, groups that had been separated from each other long enough to diverge genetically, spread across a geographic range that may have encompassed much of Asia from the Siberian taiga to the tropical islands of Southeast Asia. And that left their genetic mark in the populations of modern humans that passed through their territories at different times. In January of 2026, a team from the Max Planck Institute published the first systematic paleogenetic analysis of Denisovan DNA. segments in ancient and present-day humans, tracking these segments over the past 40,000 years across continental Eurasia. The findings sharpened the picture considerably. The earliest East Asians, represented in the genetic record by an individual called Tan Yuan, who lived in what is now China approximately 40,000 years ago, carried the highest level of Denisovan ancestry of any late Pleistocene individual examined, over 0.2% of their nuclear genome. That ancestry subsequently declined in East Asian populations over the following tens of thousands of years, diluted by gene flow from populations carrying less Denisovan DNA. The Denisovan ancestry present in West Eurasia today arrived there mainly through migrations from Eastern Eurasia after 12,000 years ago. The distribution of Denisovan genetic legacy in the modern world is not a static map. It is a record of movement, mixture, and change, still being written by the demographic processes of the past 100 centuries. But genetics, however powerful, tells you about molecules. It does not tell you about faces. It does not tell you about how these people stood or moved, or how they appeared to the modern humans who met them. For 15 years after the discovery of the Denisovans, we had no answer to the simplest question you could ask about another person. What did they look like? The answer, when it began to emerge, came not from Denisova Cave at all. It came from a riverbed in China. In 1983, a large and unusual skull was found by a construction worker in Harbin, in the Heilongjiang province of northeastern China. It was an impressive specimen. The cranium was large, with massive brow ridges and a wide face, clearly archaic in its anatomy, but with a brain case volume within the modern human range. The worker hid it in a well, reportedly to keep it away from Japanese occupying forces. It stayed there for nearly 90 years. In 2018, an elderly man on his deathbed told his family where it was. The skull was recovered, donated to Hebei GEO University, and scientists began analyzing it. It was named the Harbin skull, the Dragon Man, and dated to approximately 146,000 years ago. When the morphological analysis was published in 2021, several researchers argued that the Harbin skull represented a new species of Homo longi, and that this species was the closest known relative of modern humans. But the identification was morphological only. No DNA had been recovered from the specimen, and the species designation remained contested. Then, in 2025, two independent research groups published papers that changed the Harbin skull's status fundamentally. One team, led by paleogeneticist Xiaomei Fu, had extracted proteins from the skull after previous attempts to recover DNA had failed. Proteins survive longer than DNA under many preservation conditions, and their sequences carry phylogenetic information, though with less resolution than full genomic DNA. The protein analysis of the Harbin skull revealed 3 variants that are unique to Denisovans: amino acid substitutions in structural proteins that had been identified in the Denisova cave specimens and that do not appear in either modern humans or Neanderthals. A second supporting study isolated mitochondrial DNA from ancient dental plaque recovered from the skull's teeth. and the sequence was consistent with the Denisovan identification. The conclusion, now supported by two independent molecular lines of evidence, was that the dragon man, the Harbin skull, was a Denisovan, a Denisovan who had lived in northeastern China 146,000 years ago, who had stood, presumably, about 6 feet tall, who had a face. That face is not a modern human face. The brow ridges are massive, projecting forward over deep-set eye sockets. The cheekbones are wide and flat. The jaw is robust, but the cranium is capacious. The brain inside it was large, within the range of our own. When you look at a reconstruction of the Harbin skull, you are looking at someone who would have registered to our ancestors as unambiguously a person. Powerfully built, differently proportioned, unmistakably archaic in some features, but with eyes that held a volume of grey matter comparable to your own. Whatever intelligence means in the context of a species we cannot directly observe, this was not an animal. This was a mind. A different kind of mind from ours, but a mind. And now, in October of 2025, the picture of Denisovan biology deepened in a way that no one anticipated. A team at the Max Planck Institute generated the high-quality genome assembly of a second Denisovan individual, a man who lived in Denisova Cave approximately 200,000 years ago, making him the oldest individual of any human species whose genome has been sequenced to high coverage. This is more than twice as old as the previously sequenced Denisovan specimen. And what his genome contained was, to put it mildly, unexpected. The genome of this 200,000-year-old Denisovan male showed clear evidence that his ancestors had interbred with early Neanderthals. This was not new. Previous analysis had shown Denisovan-Neanderthal admixture. But it also showed traces of ancestry from a third group that could not be matched to any known human species. Not modern human, not Neanderthal, not the Denisovans as previously characterized. Something older. The researchers described it as a super-archaic ghost lineage, a population that diverged from all other known human groups before the split between Denisovans and Neanderthals, that persisted long enough to interbreed with the ancestors of the 200,000-year-old Denisovan from Denisova Cave, and then disappeared from the record without leaving any identified fossil or DNA sample that can be matched to it. A ghost inside a ghost. We found a species we didn't know existed by sequencing the DNA of a species we discovered 15 years ago from a finger bone. Human evolutionary history is not a tree. It is a network, a tangled, recombinant, constantly surprising network of populations that met and mixed and separated and mixed again across a landscape that we are only beginning to map. And then, in May of 2026, literally days before this recording, another piece of the puzzle arrived from an unexpected direction. Researchers analyzing teeth, approximately 400,000 years old from multiple Chinese archaeological sites, found a protein variant in those specimens, a specific amino acid substitution in a structural protein, the same variant that appears in Denisovans. The researchers' interpretation was that this variant most likely originated in populations related to Homo erectus, who occupied much of Asia for more than 1 million years before later appearing in Denisovans. The implication is that Denisovans did not simply emerge from nowhere in the Altai and spread eastward. Their origins reach back through Homo erectus populations that had been in Asia for hundreds of thousands of years, before Denisovans as a distinct group can be recognized. The Denisovan lineage is not a branch that appeared suddenly in the late Pleistocene. It is a continuation of something much older, rooted in the long Asian presence of our genus, shaped by 1 million years of evolution on a continent that modern humans were only beginning to enter. None of this, of course, explains where the Denisovans actually lived across their apparent geographic range. The genetic evidence implies a distribution spanning from the Siberian taiga to the tropical islands of Southeast Asia, an environmental breadth that would make them one of the most ecologically flexible hominin species known. The Tibetan plateau evidence is particularly striking in this regard. The high-altitude gene EPAS1, which regulates the body's response to low oxygen levels and is responsible for the remarkable altitude tolerance of modern Tibetans, was acquired by the Tibetan population from Denisovans through ancient interbreeding. This gene variant allows Tibetan populations to live and work at altitudes above 14,000 feet, with physiological efficiency that other human populations cannot match. It appears to have entered the modern human gene pool approximately 30 to 40,000 years ago, and it has since been under strong positive selection in Tibetan populations, meaning that people who carried it survived and reproduced more successfully than people who did not, and over generations it spread through the population. A Denisovan gene conquered the roof of the world, and it is still there in the bodies of millions of people living at altitude, doing exactly what it evolved to do in whatever high-elevation environment the Denisovans who originally carried it called home. The geographic picture assembled from these multiple lines of evidence, the Altai cave specimens, the Harbin skull, the Penghu mandible from the Taiwan Strait identified as Denisovan through protein analysis in 2025, the EPAS1 gene in Tibetans, the admixture signal in Melanesian and Australian genomes, is a picture of a species or a complex of related species that occupied an enormous swath of Asia for hundreds of thousands of years. They did not occupy it as a uniform population. They were differentiated, regional, adapted to local conditions in ways that left genetic signatures in the modern humans who passed through their territories. They were, in the language of ecology, a successful and widespread genus. They dominated Asian hominin diversity for a period of time that dwarfs the entire duration of recorded human history. And then they vanished. Or rather, they did not entirely vanish, because the vanishing is not quite the right description of what happened. Here is where the story arrives at something philosophically significant. The question of what happened to the Denisovans is usually framed as an extinction question, and in the narrow sense, it is. There are no Denisovans today. No population that we can identify as carrying primarily Denisovan ancestry. No community somewhere in the forests of Borneo or the highlands of New Guinea that represents an unbroken line of descent from the people who left those molars in Denisova cave. In that sense, they are gone. But in another sense, the sense that the geneticists who study ancient admixture find increasingly important, they are not gone at all. They are present in the genomes of billions of living people, not as a trivial residue, but as functional genetic material that is actively doing things in the bodies of the people who carry it. The MUC19 gene is a striking example of this. Published in August of 2025 by a team at the University of Colorado Boulder, the study showed that this gene, inherited from Denisovans through ancient interbreeding, is present in modern Latin Americans with Indigenous American ancestry and in DNA recovered from individuals at archaeological sites across North and South America. The frequency at which the gene appears in modern human populations suggests it was under significant natural selection, meaning it provided a survival or reproductive advantage to the people who carried it. The gene is involved in immune processes, suggesting that it may have helped early American populations fight off pathogens encountered as they migrated into a new continent. People crossing the Bering Land Bridge into the Americas, perhaps 15 to 20,000 years ago, carried Denisovan DNA with them. And when they arrived in the Americas and encountered the disease environments of a new world, that Denisovan gene may have helped them survive. A species that was already extinct, or at least no longer recognizable as a distinct population, was still actively contributing to the survival of people on the other side of the planet. This is the deepest implication of the Denisovan story. Human evolution is not a relay race in which one species runs its leg and hands the baton to the next. It is something messier and more interesting. It is a process in which populations meet and exchange genetic material, in which useful innovations developed by one group become available to other groups through interbreeding, in which the biological legacy of a vanished species continues to operate in the bodies of its survivors' descendants thousands of generations later. The Denisovans are a case study in this process at its most dramatic. A species known entirely through its molecular traces in other species, whose contributions to modern human biology are now being systematically catalogued, and whose geographic and temporal range, as of 2026, is still being mapped with new data arriving every few months. The question of why the Denisovans disappeared, or why, more precisely, they are no longer recognizable as a distinct population, does not have a single definitive answer. The demographic modeling suggests that it was not a catastrophic extinction event of the kind that ended the non-avian dinosaurs. It was more likely a gradual process of population dilution, in which incoming modern humans, expanding across Asia from the west, and possibly interacting repeatedly with local Denisovan groups, eventually outnumbered them to the point where the Denisovan genetic contribution, while real and consequential, became a minority fraction of the ancestry of succeeding generations. On this model, the Denisovans did not die out so much as they were absorbed, incorporated genetically into the expanding modern human populations that replaced them as the dominant hominin of Asia. This is not a comforting narrative, and it should not be presented as one. The process by which one human population absorbs another, however genetically cooperative in its molecular outcomes, is rarely a gentle story at the level of the individuals involved. But it is what the genetic evidence describes. Now let me tell you what I think about all of this. Because the Denisovan story, more than almost any other discovery in recent science, forces a kind of reckoning with our assumptions about what makes us human and what makes us alone. The dominant popular narrative of human evolution has always had a competitive, zero-sum quality to it. One species replaces another. The smarter, or the more numerous, or the more technologically sophisticated population wins. The others lose. Neanderthals and Denisovans and all the other archaic humans are gone because we replaced them, because Homo sapiens was simply better in some fundamental way at the business of being a large-brained primate in the Pleistocene. This narrative is satisfying, it is clean, and it is substantially wrong. What the genetics of the past decade has revealed with increasing clarity is that the transition from a world of multiple human species to a world with one was not primarily a story of replacement. It was a story of absorption, of contact and mixture, and the incorporation of archaic genetic material into modern human populations across multiple events on multiple continents over a period spanning tens of thousands of years. Modern humans did not emerge from Africa as a biologically complete product and then sweep aside everything in their path. They emerged as a population, interacted with other populations, and became what we are through a process of genetic exchange that involved taking useful things, immune genes, altitude adaptations, cold tolerance mechanisms, from the people they replaced. We are not the victors of an evolutionary competition. We are a mosaic, a composite assembled from multiple ancestral populations, including populations that no longer exist as distinct entities, but whose contributions are woven through the biological fabric of every living person on Earth. The Denisovans are the most dramatic example of this, because they are the most recently discovered, and because the discrepancy between how little physical evidence we have of them and how much genetic evidence is uniquely striking. But they are not an anomaly. They are a pattern that tells us something important about the nature of our species and the nature of evolution itself. Evolution does not select for purity. It selects for function. A gene that helps you survive at altitude does not care whether it originated in a Denisovan or a modern human. A gene that helps your immune system recognize a new pathogen does not carry a flag identifying its ancestral source. What natural selection sees is the phenotype, the effect on the organism's survival and reproduction. And what it does with useful variants is spread them through the population regardless of where they came from. The Denisovans provided some useful variants. Those variants are now in billions of human bodies, doing useful things, with no memory of and no obligation to the species that developed them. This is how evolution works. It is, in its way, both completely impersonal and completely extraordinary. What I find most striking after following this story for years is not the genetics. The genetics are extraordinary, but they are ultimately a technical achievement, the result of the development and application of increasingly sophisticated analytical tools to ancient biological material. What strikes me is the phenomenological fact. The fact that somewhere in Asia, between 40,000 and 80,000 years ago, a Homo sapiens and a Denisovan looked at each other across whatever distance separated them, and what happened next was not purely conflict or competition. Something else happened, something that left a trace we can still read today. We do not know the language they spoke, or whether they shared any language at all. We do not know the context, whether this was a single individual encounter or a prolonged period of contact between communities. We do not know how it felt to them on either side of that encounter, but we know the outcome. They had children together, and those children had children. And those children eventually became people who crossed a land bridge into a new continent, carrying with them in their cells a fragment of DNA from a species that would eventually be discovered from a finger bone found in a cave in Siberia 15 years ago by researchers who had no idea what they were about to find. The Denisovans are gone, and they are everywhere. Every person reading this with ancestry from East Asia, Southeast Asia, Oceania, or the Americas is, in a small but real sense, partly Denisovan. The species that we discovered from a fragment of a child's finger is the species that helped shape what we became. That is their story. And as long as the people who carry their genes are alive, it is not entirely over. The discovery that Denisovan DNA was present in Melanesian and Australian genomes immediately raised a problem of geography that has occupied researchers ever since. If the only known Denisovan fossil was from Siberia, how did Denisovans come to be present in Southeast Asia at the time when the ancestors of Melanesians and Australians were passing through? The answer, which has emerged gradually over the past decade and sharpened considerably in 2025 and 2026, is that the Denisovans were not a Siberian species with occasional forays south. They were an Asian species in the fullest sense, spread across an enormous geographic range, differentiated into regional populations, adapted to environments ranging from the subarctic taiga to the tropical coastlines of what is now Indonesia, and present across this range for a span of time that makes their eventual disappearance all the more striking. The evidence for this geographic breadth comes from multiple directions. The most dramatic piece is from Tibet. The Tibetan plateau is one of the most physiologically demanding environments on Earth. A vast elevated landscape sitting on average at more than 14,000 feet above sea level, where the partial pressure of oxygen is roughly 60% of what it is at sea level. Most human populations that move to high altitude suffer from a condition called chronic mountain sickness, in which the body's attempt to compensate for low oxygen by producing more red blood cells becomes pathological, thickening the blood to dangerous levels. Tibetan populations do not suffer from this condition to the same degree. Their bodies handle altitude with a physiological efficiency that has long puzzled researchers because the relevant genetic variants, specifically variants of a gene called EPAS1, which regulates the body's response to low oxygen, are vanishingly rare in other human populations, but extremely common in Tibetans. In 2014, researchers discovered why. The Tibetan EPAS1 variant was not a mutation that arose independently in the Tibetan population. It was inherited from the Denisovans. At some point in the past 30 to 40,000 years, ancestors of the modern Tibetan population interbred with a Denisovan group that had been living at high altitude in Central Asia, long enough to have evolved effective physiological adaptations to low oxygen conditions. The gene passed into the modern human population was subject to strong positive natural selection because the people who carried it survived and reproduced more successfully at altitude and is now present in roughly 80% of Tibetans. A Denisovan adaptation conquered the highest plateau on Earth, and it is still there, doing its job in the lungs and bone marrow of millions of people whose ancestors acquired it from a species that no longer exists as a distinct population. The EPAS1 story tells us something important about where at least some Denisovans lived. They were not confined to the lowland forests of Southeast Asia. They were in the mountains, possibly at very high altitude, possibly for long enough to have evolved specific physiological adaptations to that environment. This is a species capable of inhabiting the ecological extremes of an enormous continent, and the genetics suggests that different Denisovan populations had been separated from each other long enough to diverge genetically, meaning the species had been spread across Asia for a substantial period of time before modern humans arrived to disrupt the picture. Now here is the first genuinely unexpected turn. The genetics of Denisovan ancestry in modern humans does not tell a story of one interbreeding event. It tells a story of several, and the Denisovan populations involved in those events appear to have been genetically distinct from each other, different enough that researchers can distinguish their contributions in the DNA of their modern human descendants. The Denisovan ancestry in Papuan and Australian populations is genetically different in measurable ways from the Denisovan ancestry in East Asian populations. The EPAS1 gene that Tibetans carry represents a third Denisovan contribution from what appears to have been a population distinct from both of the others. In January of 2026, a team at the Max Planck Institute published the first systematic paleogenetic analysis tracking Denisovan DNA segments over the past 40,000 years across continental Eurasia, using a compilation of published ancient genomes from across the region. Their findings confirmed and extended this picture of multiple distinct Denisovan populations. The earliest East Asians in the dataset, represented by an individual called Tian Yuan, who lived in what is now China, approximately 40,000 years ago, carried the highest level of Denisovan ancestry of any late Pleistocene individual examined, over 0.2% of their nuclear genome. That ancestry declined in East Asian populations over subsequent millennia, diluted by gene flow from populations carrying less Denisovan DNA. The Denisovan genetic contribution present in West Eurasia today arrived there mainly through migrations from Eastern Eurasia after 12,000 years ago, carried westward by the demographic movements of the early Holocene, long after the Denisovans themselves were gone. What this means is that the Denisovans were not a single population that modern humans encountered once and absorbed. They were a family of related populations spread across Asia, each carrying a somewhat different genetic profile, each contributing to the ancestry of the modern humans who passed through their territories. We did not meet the Denisovans. We met the Denisovans plural, a geographically distributed, genetically diverse constellation of human groups that occupied the largest continent on Earth for hundreds of thousands of years before modern humans arrived and began the process that would eventually leave them as a genetic memory rather than a living presence. And then, in 2025, we finally found their face. The story of the Dragon Man begins in 1983, when a construction worker widening a bridge over the Songhua River in Harbin, in Heilongjiang province in northeastern China, found a large and unusual skull in the riverbed sediment. It was a massive specimen, the cranium large and robust, the brow ridges projecting heavily over deep-set eye sockets, the face wide and the jaw powerfully built. But the brain case was capacious. The brain volume was within the range of modern humans. The worker, reportedly concerned about Japanese occupying forces in the region, hid the skull in an abandoned well. It stayed there for 85 years. In 2018, on his deathbed, he told his family where it was. The skull was recovered, donated to Hebei GEO University, and scientific analysis began. Initial morphological analysis published in 2021 suggested this was a new species, Homo longi, and argued it was the closest known relative of modern humans, more closely related to us than Neanderthals were. But the identification was morphological only. No DNA had been recovered. The species designation was contested. The skull sat in an uncertain taxonomic position, known to be important and argued about, but not definitively placed. Then in 2025, two independent research teams resolved the question through molecular evidence. The first, led by paleogeneticist Qiaomei Fu, the same researcher who had helped establish the Denisovan identity of the original Denisova cave specimens, extracted proteins from the Harbin's skull. Proteins survive longer than DNA under many preservation conditions, and the amino acid sequences they contain carry phylogenetic information, though with less resolution than full genomic data. The protein analysis of the Harbin skull revealed 3 amino acid variants unique to Denisovans, substitutions in structural proteins that appear in the Denisova cave specimens, and nowhere else in the existing database of ancient and modern human proteins. A second independent team isolated mitochondrial DNA from ancient dental plaque recovered from the skull's teeth. The sequence was consistent with Denisovan identity. Two independent molecular lines of evidence from two different research groups using two different types of biological molecules reached the same conclusion. The Dragon Man was a Denisovan who lived 146,000 years ago in northeastern China, who stood by the proportions of the skull as a large and powerfully built individual, who had a face that would have been unmistakably hominin to anyone who looked at it. Massive brow ridges, wide cheekbones, the deep eye sockets of an archaic human anatomy, but whose cranial capacity fell within the modern human range. This was not a brute. This was a being with a large brain, living in the temperate forests and river valleys of Pleistocene northeastern China, part of a species that we now know extended from that latitude all the way south to the tropical waters of the Taiwan Strait. Because also in 2025, a mandible, a lower jaw, that had been recovered from the seafloor of the Penghu Channel off the coast of Taiwan by fishermen's nets in the early 20th century, and then sat in a museum collection for decades, was reanalyzed using protein extraction techniques. The Penghu 1 mandible was identified as Denisovan. The geographic picture expanded again. The Denisovans were not a northern Asian species. They occupied both the cold highlands of Siberia and Tibet, and the temperate forests of northeastern China, and the subtropical coastlines where Taiwan now sits above the submerged land that connected it to the Asian mainland during periods of lower sea level. Now the story delivers its deepest surprise. In October of 2025, researchers at the Max Planck Institute published the high-coverage genome of a Denisovan individual designated Denisova 25, a man who lived in Denisova Cave approximately 200,000 years ago. This made him the oldest individual of any human species whose genome has been sequenced to high quality, more than twice as old as the previously sequenced Denisovan specimen. And what his genome contained pushed the boundaries of what we thought we knew about the complexity of Pleistocene human diversity. The genome of this 200,000-year-old Denisovan showed clear evidence that his ancestors had interbred with early Neanderthals. This had been established by earlier work on Denisovan genetics. But it also showed traces of ancestry from a group that could not be matched to any known human species in the existing database. Not modern human, not Neanderthal, not the Denisovans as previously characterized from younger specimens. Something older. The researchers described it as a super-archaic ghost lineage, a population that had diverged from all other known human groups before the split between Denisovans and Neanderthals themselves, that had persisted long enough to interbreed with the ancestors of Denisova 25 approximately 200,000 years ago, and that then disappeared from the record without leaving any identified fossil or ancient DNA sample that can be matched to it with current data. Let the implication of that settle. We found evidence of a previously unknown human species, not by discovering its fossils, but by finding its DNA inside the genome of a specimen belonging to a species that was itself only discovered 15 years ago from a different piece of ancient DNA. We are finding ghosts inside ghosts. Human evolutionary history is not a tree with a few well-defined branches. It is a network, dense, recombinant, populated with lineages that we are only beginning to detect, and that we may never fully characterize because the physical evidence of their existence has not survived in the geological record in recoverable form. And then, in May of 2026, days before this recording was made, another piece arrived. Researchers analyzing teeth approximately 400,000 years old from multiple Chinese archaeological sites found a specific protein variant in those specimens. The same amino acid substitution that appears in Denisovans. The researchers' interpretation was that this variant most likely originated in populations related to Homo erectus, the species that occupied much of Asia for over 1 million years before the appearance of either Denisovans or Neanderthals, and that it later appeared in Denisovans, implying a direct biological connection between the long-resident Asian populations of Homo erectus and the Denisovan lineage that eventually emerged from them. The Denisovans did not parachute into Asia from some external origin. They grew from it. Their roots reach back through the deep Asian hominin record, through Homo erectus populations that had been adapting to Asian environments for 1 million years before Denisovans as a recognizable genetic entity can be detected. The Denisovan lineage is the continuation of something ancient and continuous, the long Asian branch of our genus, shaped by 1 million years of isolation and local adaptation on the world's largest landmass. This brings us to the question that sits at the end of all this evidence, the question that the genetics raises and that no amount of ancient DNA can fully answer. What was it like to be a Denisovan? Not in the trivial sense of daily life, what they ate, how they made tools, how they organized their social groups, though these are interesting questions. In the deeper sense, what kind of minds were these? What did they know, and how did they know it? When a Denisovan and a modern human met in the forests of Southeast Asia 60,000 years ago, what did each of them see in the other? We cannot answer this from the evidence we have. The Denisovans left no art that we have identified, no objects whose function was clearly symbolic rather than utilitarian, no burials with grave goods. This absence does not prove the absence of symbolic behavior. The Neanderthals were long assumed to be incapable of symbolic thought before evidence to the contrary accumulated, and the Denisovans have left us far fewer specimens to examine. The absence of evidence is not evidence of absence, particularly in a species known from so few physical remains. What the brain size of the Harbin skull suggests is that the Denisovans had the neural hardware for cognitive complexity comparable to our own. What they did with it is a question we cannot yet answer. What we can say is this: the Denisovans survived as a distinct population for at least 200,000 years in Asia, possibly considerably longer if we count their Homo erectus ancestry as continuous with their lineage. They adapted to environments ranging from subarctic Siberia to tropical Southeast Asia. They developed at least one physiological adaptation. the EPAS1 variant, that was sophisticated enough to be maintained by natural selection in modern human populations for tens of thousands of years after the Denisovans themselves disappeared as a distinct group. They were not a failed experiment. They were a successful one. A species that occupied and adapted to a continent for geological spans of time, that interacted with multiple other human species, including the mysterious super-archaic ghost lineage whose identity we still do not know, and that eventually met modern humans and contributed to what those modern humans became. The end of the Denisovans, like the end of the Neanderthals, was almost certainly not dramatic. It was not a battle, not a plague, not a climate catastrophe that wiped them out in a geological instant. The most likely scenario, as best as the demographic genetics can reconstruct it, is a gradual process of population replacement and absorption. Modern humans, expanding across Asia from the west, were more numerous and possibly more demographically flexible than the Denisovan populations they encountered. Each interaction, each period of interbreeding, each exchange of genetic material left the Denisovans a somewhat smaller proportion of the ancestry of subsequent generations. Over time, measured in thousands of years rather than centuries, the Denisovan genetic contribution shrank from a major component of the ancestry of Asian populations to a minor one, and eventually to the fragments, ranging from a fraction of a percent to several percent depending on the population, that persist today. But those fragments are not trivial. And this is the final, most important point about the Denisovan story, as we understand it in 2026. The MUC19 gene, published in August of 2025 by researchers at the University of Colorado Boulder, is present in modern Latin Americans with Indigenous American ancestry and in ancient DNA from archaeological sites across North and South America. Its frequency in these populations indicates it was subject to positive natural selection, that the people who carried it survived and reproduced more successfully than those who did not. The gene is involved in immune processes, and the most likely interpretation is that it provided some advantage against pathogens encountered as the ancestors of modern Native Americans migrated into the Americas, crossing the Bering Land Bridge approximately 15 to 20,000 years ago, and entering a continent whose disease environment was unlike anything they had encountered in Eurasia. A Denisovan immune gene helped people survive in the Americas. That gene is still in the bodies of millions of living people. It is still doing its job. The EPAS1 gene is still helping Tibetans breathe at altitude. The various immune and metabolic variants inherited from Denisovans in Southeast Asian and Melanesian populations are still present and presumably still functional. The Denisovan contribution to modern human biology did not freeze in place at the moment of interbreeding. It entered a living, changing pool of genetic material and has been subject to the same evolutionary pressures as every other part of the human genome. Some Denisovan variants were neutral and drifted through populations by chance. Some were mildly deleterious and were slowly purged by natural selection. And some, the EPAS ones, and MUC19S were genuinely useful in the environments where the people who carried them lived, and were amplified by selection until they became common. The Denisovans did not simply give us their genes. They gave us genes that helped us conquer environments they themselves had already conquered. They showed us, genetically, how to survive in Asia, and we used that knowledge in ways they could not have anticipated, carrying it to Tibet and Melanesia, and eventually to the Americas, where it is still working in the immune systems of people whose existence would have been inconceivable to the individual who first developed that genetic variant in an Asian highland 100,000 years ago. This is what I think about when I think about the Denisovans. Not their disappearance, their continuation. The popular framing of the archaic human story is almost always elegiac. We replaced them. We were better. They are gone. And there is something to that framing. The demographic reality is that Denisovans as a distinct population no longer exist. And whatever was specifically Denisovan about their way of being in the world, their languages, their social structures, their relationship to the landscapes they inhabited, whatever symbolic or spiritual life they may have had, is gone with them, unrecoverable. That loss is real. The disappearance of any human population, any way of being human, is a kind of diminishment of the total range of human possibility. But the framing misses something essential. The Denisovans are not gone in the way that a species goes extinct when the last individual dies and the lineage ends. They are gone in the way that a river is gone when it joins a larger river, absorbed, transformed, continuing in a different form, contributing to something that could not have been what it is without that contribution. The modern human populations of Asia and the Americas are not replacements for the Denisovans. They are, in a measurable and specific biological sense, their inheritors. Every person with Tibetan, Melanesian, Aboriginal Australian, or Indigenous American ancestry carries a fragment of Denisovan biology. That biology is active. It is doing things. It is part of what makes those populations what they are. We found the Denisovans 15 years ago from a fragment of bone small enough to hold between 2 fingers. In the years since, we have found their face, their geographic range, their physiological contributions to living human populations, their relationship to species we did not know existed, and their roots in the deep hominin history of Asia. Every year has brought something new. The story is not finished. It may not be finished for decades. More ancient genomes will be sequenced. More protein analyses will identify Denisovan individuals among the thousands of hominin fossils currently sitting unanalyzed in museum collections across Asia. More living genetic variants will be traced back as to their Denisovan source. What we already know is enough to permanently alter the picture of human evolution. We did not replace the other humans. We incorporated them. We are a mosaic assembled from multiple ancestral populations, each contributing something to the whole, each leaving a trace in the genome that natural selection has been sorting through ever since. The Denisovans are the most dramatic example of this process because they are the most recently discovered and the most molecularly present. But they are a pattern, not an exception. And the pattern tells us something important about what we are. We are not the victors of an evolutionary competition. We are a collaboration, complex, unplanned, spanning hundreds of thousands of years and 2 continents. A collaboration between species who never knew they were collaborating, whose interaction was almost certainly not experienced by any of the individuals involved as anything so grand or so purposeful, but whose outcome, seen from the distance of millennia and read through the lens of molecular genetics, has exactly that quality. We became what we are because of who we met, and the Denisovans, more than anyone else we have yet found, changed the meeting. They are gone, and they are in you. That is not a metaphor. That is a fact written in the molecule that carries everything worth carrying forward, the record of every adaptation that worked, every encounter that produced survivors, every generation that made it through to the next. The Denisovans made it through, not as a species, as a contribution, and in the bodies of the people who carry their genes, doing the quiet, essential work of survival, their story continues.